Good evening or good afternoon, everybody. Welcome to this ATS Pulmonary Circulation webinar on the best of ATS with abstracts and presentations from the last ATS. My name is Jurjan Amon. I'm from Amsterdam, the Netherlands. I'm together with Roberto Bernardo. We are hosting this session. We have an exciting, a number of exciting talks. Ranging from interstitial macrophages to parasites, cardiomyocytes, and actually covering the whole biology of pulmonary hypertension with four speakers. Dr. Kumar from UCSF, Dr. James from Indiana University, Dr. Paul Yu from Harvard University, and Dr. Sugu from Anschutz University. And the discussion is moderated by, we have comments from a number of panelists who include Professor De Man from Amsterdam, Professor Soni Pulamsetti from Gießen, Germany, and Professor Soban Umar from UCLA, United States. So I'm looking forward for a very exciting session. And I would like to give the word first to Rahul Kumar, who will speak about crosstalk between recruited and resident interstitial macrophages in schistosoma-induced pulmonary hypertension. Rahul, if you start sharing your slides, the floor is yours.
Thank you.
Sorry, one thing to add. If you have any questions during the talk, please add them to the chat, and we will take care of these questions.
Sure. Thanks, Ahmed, for such a wonderful introduction. I'm Rahul Kumar from University of California, San Francisco. First of all, I'm thankful to the organizer for selecting my work for this webinar. Today, as Ahmed mentioned, I'm going to talk on crosstalk between recruited and resident macrophage in schistosoma-induced pulmonary hypertension. So our lab primarily works on schistosoma-induced pulmonary hypertension, which is considered as one of the leading cause of pulmonary hypertension worldwide. And we are trying to understand the pathogenesis, and our focus is to understand how the immune cells causes pulmonary hypertension. So this is considered like second most tropical neglected disease, which is endemic in more than 90 countries, majorly in African regions, and some part of South America and China. So it has been shown that even in the clinical setting, there are three species which is commonly caused pulmonary hypertension, which includes the Schistosoma mansoni, which is the major primary species that cause pulmonary hypertension, and the second is Schistosoma japonicum. And mansoni and japonicum, they live in the portal system. Their primary target organ is like hepatointestinal region, and there's another species called Schistosoma hematobium, which infects the urinary bladder. So as per World Health Organization, it has been classified under group 1 pH based on their clinical presentation and the mechanism involved in the pathogenesis of the disease. And as I mentioned, that this is considered as the leading cause of pulmonary hypertension worldwide. And in almost around more than 200 million people are infected with schistosomiasis, where all these three parasites are majorly involved. And approximately 2 million people are affected with pulmonary hypertension, where 60% is contributed from Schistosoma mansoni, 30% by parasite called hematobium, and 10% by Schistosoma japonicum. So to understand the pathogenesis, like how the inflammatory immune cells play a role in driving this inflammatory response, which leads to vascular remodeling and ultimately pulmonary hypertension. So we created a model in our lab, which is highly reproducible, and this is basically an inflammation-based model, where we first sensitize mice with the schistosoma egg. So we, as here you can see, there are like different structure morphology of these eggs. This is japonicum, which is like a round shape, and these two, hematobium and Schistosoma mansoni, they are like oval shape. They have some spike region here, which goes and stick to the tissue. So here we created this. We first sensitize mouse with this species, and then 2 weeks later we gave intravenous challenge with this species, and the species, this eggs we get we isolate from the infected mice that we get from the Biomedical Research Institute, NIH, and we harvest these eggs from the liver and the urinary bladder, species Japonicum, and then we perform this we create this 21 days model where we first sensitize, we challenge after 2 weeks, and after 7 days of challenge, we perform right heart cath, collect tissue to do downstream analysis. So here you can see that when we sensitize mice with Schistosoma mansoni and challenge with Schistosoma mansoni egg, it causes severe pulmonary hypertension. This is done by the right heart catheterization. We perform the right heart catheterization and compared to unexposed mice. Schistosoma japonicum and Schistosoma hematobium, they also cause pulmonary hypertension, but the severity is comparatively less compared to Schistosoma mansoni, which again goes with the literature and the reported cases in the clinical setting, where a majority of the patients who are presented with pulmonary hypertension have Schistosoma mansoni as a primary species. We and others have shown that the mouse model has shared some key clinical pathophysiology with the human disease, people who die with the Schistosoma mansoni pulmonary hypertension. The question, the overall mechanism that has been studied and known till yet by utilizing human specimen and the mouse model is that after schistosoma infection, it triggers severe TS2 inflammation, which basically the TS2 cytokine IL-4, IL-13 gets released, and these cytokines are primarily released by CD4 T cells, which is present in the lungs. And we also have reported that there's a higher level of thrombospondin 1 and TGF-beta in the lung, and we have shown as a mechanism that thrombospondin 1 is one of the known biological activator of thrombospondin 1, the TGF-beta 1, which activates TGF-beta 1 to cause pulmonary hypertension. In addition to this finding, we also found that the schistosoma infected lung has very high level of macrophage ligand, monocyte macrophage ligand CCL2, 7, and 12. And in addition to this, we have found that the infected lungs have very high number of interstitial macrophage, and we believe that the number of high interstitial macrophage is due to the recruitment of circulatory monocytes that goes into the lungs after infection. So the interstitial macrophage, which is not been very well characterized in schistosoma pulmonary hypertension. In the homeostatic setting, in the homeostatic condition in mouse model, literature have suggested the existence of 2 to 3 forms of interstitial macrophage, but nobody has looked into the inflammation, schistosoma-induced inflammation, how this macrophage like crosstalk and what are these macrophage does. So we try to characterize first the macrophage, and based on the literature, it has been shown that there are like primarily 2 types of macrophage. One is resident interstitial macrophage, and one is recruited, but nobody knows how this macrophage crosstalk. So the primary marker that like all the literature have been used to characterize the recruited versus resident macrophage is CCR2, MSC2, and FLR2, which is folate receptor, LIVE1, and TMID4. So either of these 3 is being expressed by the resident macrophage, and the recruited one express CCR2. The MSC one is something between these 2, recruited versus the resident one, which I will talk in a couple of slides. So in the current study, our main goal was to characterize the interstitial macrophage in TS2 inflammation due to schistosoma mansoni, and how these macrophages crosstalk. So as I mentioned that we see a lot of CCL2 ligands, and we believe that recruited macrophage which express CCR2, it gets recruited. So there is some sort of crosstalk between recruited versus the ligand which is present in the lungs. So to understand this, we used flow cytometry analysis. We performed single-cell RNA sequencing on those sorted interstitial macrophage. And we also do some functional studies by utilizing bone marrow chimerism study. So overall, I'm not going to detail into these steps. So overall, the way we characterize the interstitial macrophage, we just selected all the CD45 positive cells, which is a pan marker for the leukocyte, and then we selected all the macrophage based on the CD64 marker. and then we characterize them based on the CD11B, CD11C. So here all the CD11B positive and CD11C intermediate to high are interstitial macrophage, and we further characterize this based on the folate receptor antibody. So here we found two population. One is FLR2, which is resident macrophage, and these are the FLR2 negative. And when we further characterize this, we again found two populations, CCR2 positive and MSC2 high. So, and this is what more or less literature has classified three types of interstitial macrophage. But we and others have found that the MSC2 high, the CCR2, they have like 96 to 90%, 96 to 97% similarity based on their gene expression pattern. So overall, when we did the statistics by comparing the exposed versus schistosoma unexposed mouse, we found there was a significant increase in the recruited interstitial macrophage, which was CCR2 positive, and there was no difference in either MSC2 or the resident macrophages. And again, we characterized this macrophage. So here, what we did was isolated the all interstitial macrophage and we did a time course analysis for day 1, day 3, and day 7 time point to understand the dynamics of these macrophages. And we utilized 10x platform to understand and characterize these macrophages. Interestingly, here you can see which is like unexposed baseline macrophage. We've overall observed 3 forms of macrophages. We named them C1s, cluster 1, cluster 2, and cluster 3. And when we give the schistosoma by challenge the mice with the schistosoma parasite, and we followed their lungs at day 1, day 3, and day 7 time point, we again found there's a huge recruitment in C1 population, which were after schistosoma exposure. And when we characterize this population, you can clearly see that that there is the C1, which we believe is the recruited macrophage. They have a distinct marker, and here we can the way why we are saying this is a recruited. They express high amount of Ly6C and CCR2 positive. And again, the MSC1, which is if you look at closely, you can see they express all the major histocompatibly complex markers. So this population C2, we believe this is the MSC2 high the interstitial macrophage. And this population, which is the resident, the FLR2 positive, we call it FLR2 positive, they express the unique markers, which is specifically expressed by these resident macrophages. So overall, we found that after the schistosoma exposure, the number of recruited macrophage increases, and it matches with what we have found in the flow cytometry analysis. Again, to confirm this, we use the double reporter mouse, which is GFP and RFP positive, to track these macrophages. So after schistosoma exposure, we found that the overall in the blood, the number of monocytes were higher. And when we characterized the same mouse lung, we found the number of CCR2 positive macrophages was significantly higher. So it indicated that these CCR2 positive macrophage gets recruited, and the number goes higher. So recently we did the lineage tracing experiment, which I'm not showing here. Again, that confirmed that this by using the lineage tracing experiment, the majority of the CCR2 positive macrophages are coming from the circulation, and that gets recruited after schistosoma infection. Whereas the resident macrophage, they are the resident origin. They originate from the non-recruited source. So then next the question was to what are the mechanism by which these two macrophage crosstalk. So here we hypothesize that after schistosoma infection, it triggers Th2 inflammatory response and produce IL-4, IL-13, and IL-4, IL-13 somehow induces the production of CCL2, 7, and 12 ligand from the resident macrophage, and this resident macrophage sends signal to the blood, and this CCR2 positive interstitial macrophage gets recruited into the lungs, where it makes thrombospondin 1, which activates TGF-beta to cause pulmonary hypertension. So first, to understand what are the source of CCL2 in the lung, we use CCL2 RFP reporter mice, and we use our same 21 days model to challenge them with schistosoma mansoni, and the unexposed mice were not sensitized or challenged with schistosoma mansoni egg. And interestingly, we found that number of CCL2 RFP positive was significantly higher in the resident macrophage. So it indicated that the number of CCL2 positive cells or CCL2 are coming from the resident macrophage. When we do the median fluorescent intensity analysis, we found that after schistosoma infection, the majority of the CCL2 are coming from the resident macrophages. And similarly, as I proposed that once these the resident macrophage, they make CCL2 and they send signal to the blood, and it recruits the CCR2 positive ions that makes thrombospondin 1. So since we don't have any reporter line for thrombospondin 1, we use wild-type mice and we perform the intracellular flow cytometry analysis. And we found that number of these thrombospondin 1 positive marker TSP recruited macrophage was positive for thrombospondin 1. And again, the median fluorescent intensity analysis showed that majority of the thrombospondin is produced by recruited macrophages. So the CCL2 is being produced by the resident macrophage, which that sends signals to the monocyte in the blood, and that gets recruited and makes thrombospondin 1. Again, this flow cytometry data was further confirmed by the single cell data, where we found that the recruited macrophage were positive for thrombospondin 1 after schistosoma exposure. And similarly, the CCL2, 7, and 12 ligands, which are being produced by resident macrophage, here you can clearly see this is highly expressed by the resident macrophage compared to other macrophage population. And then to understand the functional relevance, what we did, we blocked the CCR2 by transplanting CCR2 knockout bone marrow into the wild-type mice, and then we use our schistosoma menstrual 921 disease model, and we performed the right heart cath, and we again found that the wild-type mice that received CCR2 knockout bone marrow, so means they don't have CCR2 gene expression. So we found these mice were protected from right ventricular systolic pressure by right ventricular systolic pressure, as well as their Fulton index was the RB hypertrophy was significantly down. And similarly, when we block the thrombospondin function in this recruited macrophage by transplanting thrombospondin knockout bone marrow into the wild-type mice, and then again we used our 21 days mouse model, we found that when we block the thrombospondin function in those recruited macrophage, the monocyte, once they get recruited, they don't have thrombospondin 1, and those mouse were protected from pulmonary hypertension. And we also found that the TGF-beta signaling was also down in these mice. So overall, in the study, we have concluded that schistosoma menstrual triggers severe pH compared to other species. And our flow cytometry analysis and the single sequencing analysis revealed 3 IM subsets, and recruited CCR2 monocyte serves as the precursor of thrombospondin 1 positive IMs, and the resident, which is characterized by folate receptor that is called resident interstitial macrophage. They make CCL2 that helps in recruitment of the CCR2 positive monocytes into the lungs, and blockade of either CCR2 monocytes or its thrombospondin 1 function protects from schistosoma-induced pulmonary hypertension. So finally, I would like to acknowledge my mentor, Dr. Brian Graham, my lab mates, and our collaborators from University of Colorado and Biomedical Research Institute for providing us schistosoma-infected mice, and all the funding agencies to support this study. Thank you so much for your attention, and I'm happy to take any questions.
Thank you very much for presenting this extensive and beautiful data set. I think I will switch to the panelists. Are there any questions from the people from the panel?
Yes, I have a question. Thank you very much. It was a really clear presentation. Last PVRI webinar, Professor Tudor showed some spatial transcriptomic analysis. Is that your next step that you want to do to really understand? Because you clearly saw the CCR2 is important, and then TGF-beta, and then pulmonary hypertension, but the link with pulmonary hypertension and lung vascular remodeling is not that clear yet. So is that your next step to go into more in detail there, and how the endothelial cells and muscle cells are connecting with the CCR2 positive cells?
Yeah, thank you so much. That's very good and great question. This is what we are trying to understand. What is the crosstalk between the structural cells versus the immune cells? So now we are trying to understand how like, for example, in cystosoma mentioned, presentation of antigen is very important. And we are trying to understand like which cells, whether it is like dendritic cells or endothelial cells, are involved in presenting antigens, and then these monocytes derived macrophage gets recruited. So certainly the next step will be to understand the crosstalk between the structural versus the immune cells. And still there are a lot of study that needs to be done in this field. Still we are not trying to understand in detail about the mechanism by which this macrophage, how this gets recruited, what are the crosstalk, is there any other protein which are playing some pathological role in driving pulmonary hypertension in clinical as well as the human setting.
Yes, so do you have access to human tissue of patients?
We don't have direct access to human tissue. We have in the past, my mentor, Dr. Graham, he has collaborated in the PI in the Brazil, from where we get some human samples, and we did some staining analysis, immunostaining analysis in those tissue. But we have access to the plasma samples, blood samples, with our recent collaboration in Brazil and Zambia, so the two clinical sites. And we also have collaborated with in Brazil, where we have access to the human blood samples affected with cystosoma IPH.
Yeah, very interesting.
I know we're running behind, Georgian. Is there time for more question? Or there's a question from Zio in the chat. Maybe we can go for that, and then I'll skip my question for now. You're muted, Georgian.
Sorry. Yes, I agree. It's good to go to for the one in the chat from Zio Dai, who asked, who complimented you with the tool on the CCR2 RFP and the GFP in the CXCR3. But his question is how many RFP and GFP positive macrophage monocytes are overlapped from the CCR2 RFP CX3CR1 GFP mice?
That's a very good question. Yeah. So normally we see some. So we have not. If you look at the after cystosoma infection dynamics, they lose. We try to characterize based on the CCR2 and the CXCR1. Based on the literature, it says that in blood, the non-classical monocyte express GFP. They express CXCR1, and the classical monocyte derived macrophage they express CCR2. But if you look, but we do see some sort of overlap between these two, which we are like. We were like a little bit like it was not very clear to characterize these two populations derived from from the classical versus non-classical. So that's why we have like performed the lineage tracing experiment, where we have very like TD tomato CCR2 positive mice, where we can easily track all the mice that is coming from the recruited macrophage in cleaner picture. But we do see some overlap. I don't know how much percentage. But we have the data. Definitely, I can go back and look at the overlap between these two, GFP versus RFP. That's good. Thank you.
Perfect. Thank you very much. And I think for the sake of time, we indeed have to move to the next speaker, who's Joe James from Indiana University. And we will talk about targeting the stratophil-mediated endothelial parasite crosstalk, which ameliorates pulmonary hypertension. Joe, the floor is yours.
Yeah, hi everyone. I'm Joel from Indiana University from the Rafikova lab. And today I'd like to speak about how targeting stratophin could be a novel therapeutic strategy in pulmonary hypertension. I have no disclosures. And so recently we've been exploring mitochondrial dysfunction, and several people have been exploring mitochondrial dysfunction in pulmonary hypertension. So the general rate of pulmonary hypertension occurrence in the general population is around 15 to 30 per million. But there is one particular group of patients with a mutation in a mitochondrial iron sulfur cluster chaperone, and this protein is called NFU1 protein, and it helps inserts iron sulfur clusters into mitochondrial complexes. So patients with this mutation generally have dysfunctional iron sulfur cluster insertion, leading to highly dysfunctional mitochondria. And these patients go on to develop pulmonary hypertension. And in fact, 7 out of 10 patients with this mutation develop pulmonary hypertension. So which leads us to think that probably mitochondrial dysfunction could be a primary determinant for pulmonary hypertension. We recently published a paper in the Journal of Experimental Medicine, which explores how mitochondrial dysfunction can lead to pulmonary hypertension. So we developed a rat model with a homologous mutation in NFU1, and we see that these rats also spontaneously develop pulmonary hypertension, and they have highly remodeled vessels, which is very interesting. But how mitochondrial dysfunction leads to pulmonary hypertension is still not well explored. So enter aminopeptidase N, a protein, a very curious protein that I discovered a couple of years ago. And we found that aminopeptidase N was significantly increased in the pulmonary artery smooth muscle cells of these NFU1 rats. And curiously, we also found that patient lungs also showed an increase in aminopeptidase N expression. So what exactly does aminopeptidase N do? So aminopeptidase N, or it's also called CD13, is a protein that's involved in several cellular functions like peptide metabolism, cell motility, and also it's implicated in cancer and has been shown to increase migration and invasion. So curiously, it is also a marker of progenitor cells like pericytes. So what exactly is this protein doing in smooth muscle cells was a question that I was very curious about. So what exactly do pericytes do? They have numerous functions, and importantly, they help in capillary formation and maintenance. And pericytes are very important, and they act like progenitor cells whenever it's needed for repair. And recent papers, especially by Vinicio's group and KU1, have shown that when pericytes become dysfunctional, they dissociate from the capillaries, and they can become smooth muscle-like in nature and can cause vascular remodeling. So we see aminopeptidase N increase in the smooth muscle cells. So the question that we want to ask is, are these smooth muscle cells, do they have pericyte origins? Where exactly do these, how exactly is this APN overexpressed in the smooth muscle cells? So do we see pericyte dysfunction due to mitochondrial dysfunction is a question that I was very curious about. So what we see here, if you look at the lungs of these NFU1 pH model, we see that indeed pericytes form very poor associations with endothelial cells in the NFU1 lungs, and clearly showing that there is some sort of pericyte dysfunction. And if you look at 3D confocal imaging, we see that there is indeed poor pericyte endothelial association. And if you look at the lungs closely, at the lungs of these NFU1 rats, we see that there is highly impaired vascular development. And this could be probably pointing back to the fact that there is poor pericyte endothelial associations, and there is indeed impaired vascular development. And so what we did next was we did some single cell sequencing of the lungs. And if we closely look at just the pericytes and the endothelial cells, and we do some cell interaction analysis, we see that there is decreased pericyte endothelial associations due to mitochondrial dysfunction in pulmonary hypertension. More curiously, if you look at the smooth muscle cell population, pointed here with the arrow, you see that these smooth muscle cells seem to show increased pericyte markers as opposed to the wild type rats. And if you take, say, for example, a publicly available patient data set, we did that and we did some analysis on that. And we see that the smooth muscle cells indeed show increased pericyte markers. So the question here is, what exactly is happening to these pericytes, and how are they actually losing their phenotype, and are they becoming invasive? So looking back at our NFU1 model, if you look here, very clearly we see that the NFU1 pericytes seem to show increased canonical smooth muscle cell markers like calponin and smooth muscle myosin heavy chain. So clearly there is some sort of a phenotype shift in the pericytes, and the pericyte seems to lose their pericyte nature and become more smooth muscle cell-like in pulmonary hypertension. So the question we asked was, how exactly is this APN activated? There should be something that activates APN, right? So very curiously, I stumbled upon this protein called stratifin, and stratifin is known to be an APN regulator. And we found that it was significantly increased in mitochondrial dysfunction. So what exactly is stratifin? Stratifin comes from a family of proteins called the 1433 proteins, which are like communication signaling messengers in biological systems. And they're found throughout all organisms and all levels. And 1433 proteins are very interesting, and especially this protein called stratifin. And I found that stratifin was highly increased in the endothelial cells and also they were highly secreted by these endothelial cells. And stratifin has been shown to bind to aminopeptidase N, and it is shown to increase aminopeptidase N activity and also expression. So clearly, when we added stratifin, recombinant stratifin to pericytes, we see that the pericyte seems to become highly proliferative in nature. Very curious and very interesting. So, this is what we think is happening here. So we think that endothelial cells in mitochondrial dysfunction can secrete or overexpress stratifin, which can bind to aminopeptidase N and cause downstream signaling. Aminopeptidase N can have signaling, it has a signaling domain and can have a lot of signaling partners as well. So what we did was we developed some blocking peptides to block the signal. We disrupted the signaling of aminopeptidase N. And also we tested a peptide to block stratifin signaling. And so, when stratifin binds to APN, we think that there could be a pericyte dissociation to smooth muscle cell like cells and cause vascular remodeling. So we tried to target stratifin and see if this could reverse pulmonary hypertension in our experimental model. So as you can see here, this is just preliminary data, and we found that stratifin, blocking stratifin reverse pulmonary vascular remodeling, as you can see here very clearly. Furthermore, we found that stratifin, blocking stratifin signaling also reversed and improved RV function and also pulmonary flow parameters, which was very interesting. So this is all, we are working on this exciting data, and we did some single cell analysis as well. And as you can see here, our preliminary data shows that treating these NFU1 rats with stratifin inhibitors actually improved the pericyte endothelial association. So this is what I think is finally the overall picture of what we're trying to explore is this in a nutshell. We think that mitochondrial dysfunction can increase stratifin expression, and due to the close association of pericytes, which express APN naturally, and their association with the endothelial cells, we think that stratifin can go bind to APN and activate APN. And excessive signaling of stratifin can cause pericyte dissociation and leading to transformation into smooth muscle-like cells, causing highly remodeled arteries. And also the dissociation of pericytes from the endothelial cells can cause impaired vascular development. And this can lead to a vicious cycle in hypoxia. And we think that could be a trigger for pulmonary hypertension. So this is, we are exploring stratifin, and we're trying to see how we can actually improve pulmonary hypertension using novel peptides that we're developing to target various domains of aminopeptidase N and various ways in which we can actually block stratifin signaling. So I would like to thank the Rafikova Labs members who have helped me with this study. And also I'd like to thank Elena and Vinicio who've helped us with this study as well, and also the funding agencies. I'm open to taking questions. Thank you.
Excellent. Thank you for this very clear and wonderful story. And I want to open the floor for discussion. And I think Umar was really into a question.
Yeah, I'll be
brief
and quick, because again, I see their question in the chat. So thank you, Joel. Excellent presentation. So just quick questions. So what more do we know about stratifin? I know you're linking it to mitochondrial dysfunction, but what exactly triggers it? What's the relationship with let's say, pulmonary artery pressure overload? Have you done like any time course experiments? And are there any sex differences?
Let me first start with the sex differences. We are exploring sex differences, and we potentially see sex differences, but we're still in the preliminary stages to make a sure comment. But it's leaning towards females expressing more stratifin than males, but it's just preliminary work that we're doing right now. And what was the first question that you asked?
Is there any correlation in your model with let's say pressure overload? Have you looked at like a time course of like milder disease versus more severe disease? And And how about in humans? This
So,
peptide, yeah.
So we did study some human expression of stratifin, and we see that there is increased circulation of stratifin in general in patient plasma samples. But about your pressure question, we have not studied that yet. In time course, I have not studied any time course experiments. No.
Thank you.
I think we still have some time for a question from the chat. Roberto, I think the choice is yours.
Yeah. Nice work, Joel. Ziyuda from Arizona has a question for you. Can SFN APN signaling affect pericyte proliferation and migration in addition to transdifferentiation to smooth muscle cells?
Thank you, Zio. That's a very good question. And yes, so as I had shown the data in the slides before, when we add stratifin to pericytes, we see that pericyte proliferation increases. But I'm still studying how pericytes can transform to smooth muscle cells. There are several assays, and we are also trying to see the molecular markers of increased smooth muscle-like cell nature in pericytes when we treat them with stratifin. So this is still an ongoing study. Thank you.
Perfect. Well, there's a lively discussion in the chat. So somebody's also asking for the percentage of parasite that transforms to SMC. Maybe you can address that question in the chat so that we can move on to our next speaker, who's Paul Yu from Harvard University. And we will talk about BMP9 to regulate endothelial expression of mediators of pulmonary vascular remodeling. Paul, the floor is yours.
I think you're muted, Paul.
Thanks so much for the opportunity to present. I just wanted to point out that this work is from Ying Zhang, a very talented fellow in our lab who is not able to present herself. And these are the conflicts. So we visited this concept that dysregulated BMP and TGF-beta signaling are actually a viable paradigm for understanding signaling in pulmonary hypertension. In general, we think that this holds true. But in light of recent data with sotatercept and recent data with BMP9, wanted to make better sense of this. We know that BMP9 is absolutely a loss of function genetic risk factor for pulmonary hypertension. We've seen this across many cohorts in different ethnicities, in different countries. And there is a consensus that 1% of our group 1 population has loss of function mutations in BMP9, and these are clearly loss of function in that they inhibit signaling or inhibit expression or secretion of BMP9. But then we have, in contrast to this, the fact that BMP9 is clearly a marker for portal pulmonary hypertension, and with recent data also a marker for hepatopulmonary hypertension, hepatopulmonary syndrome. And this work done by Ivana Nikolic with Kerry Robertson, Rich Shanik, not only showed that this was a fairly unique marker of portal pulmonary hypertension among other group 1 PH, but also predicted outcomes in our group 1 pulmonary hypertension, whether or not they had portal pulmonary hypertension. But we are also familiar with this elegant work from Nick Morell's group showing that BMP9 can be administered as an exogenous protein to rescue pulmonary hypertension in several models, including genetic models, and of course, a very rigorous delayed treatment oxygen hypoxia rat pulmonary hypertension model here, where there is apparent recanalization of obliterated vessels with just the last 3 weeks of this 11-week model of treatment. And to make sense of this, this work from Mark Ormiston's group seems to support the idea that BMP9 can have different effects based on the context. And in their experiments for this ATV paper showed that wild-type endothelial cells retained an anti-proliferative function of BMP9, whereas heterozygous loss of function ECs that they engineered had a loss of that anti-proliferative function. And when they deleted both alleles of BMPR2, actually gained a pro-proliferative function. And this seems to support the notion that disease DCs tend to respond to BMP9 differently than healthy lung-derived microvascular ECs, even without BMPR2 mutations, changing their responses to BMP9 based on context. And we have this important set of data from Christophe Genebert's group with Sabine Bailey showing that BMP knockout mice are protected relatively to Sugen hypoxia and hypoxia-induced pulmonary hypertension. And this can be replicated by administering an anti-BMP9 antibody. And also in the monocrotal in rats and the Sugen hypoxia-treated rats, delayed administration of a combined BMP9/10 trap made from the ALK1 extracellular domain expressed as an Fc fusion. This ALK1 Fc can markedly reduce evidence of pulmonary hypertension and RV hypertrophy in these models. And we sought to replicate that data, and we're happy to say that this holds true not only when given in a delayed fashion, as in Christophe and Sabine's experiments, but holds true even if you give ALK1 Fc prior to the administration of Sugen and hypoxia. So the hypothesis of the experiment was that inhibiting BMP9 and 10 might be therapeutically beneficial after established pulmonary hypertension. We asked if administering before the vascular injury might be different in its effect. And it turns out that it's because it's only 2 doses at the beginning of this model as opposed to 6 doses after established disease. It's trending towards helpful, but it certainly doesn't exacerbate this disease. We did a different experiment where we thought if ALK1 Fc, which was originally developed as an anti-angiogenic drug for solid tumors, if that might replicate the anti-angiogenic signal of a VEGF inhibitor like Sugen 5416. And so in this experiment, we compared the effects of 3 weeks of hypoxia versus Sugen hypoxia versus giving ALK1 instead of Sugen in combination with hypoxia. And we hypothesized that maybe this kind of combined injury with hypoxia and BMP9/10 suppression might be injurious, but it turns out to also be therapeutic and pretty much normalizes the hypoxic-induced pulmonary hypertension that you'd see in this model. So in every context, whether we gave ALK1 Fc before, during, or after vascular injury, it seemed to be protective against pulmonary hypertension. And the effects were very comparable to other molecules that we had looked at recently. The effects of inhibiting BMP9 with a commercial antibody, this is MAV3209 from R&D, mimics the activity of not only Actor2A-Fc, the preclinical sotatercept-like molecule, but also replicates the effect of inhibiting BMP9 and 10. So inhibiting 9 is fairly similar to inhibiting 9 and 10, and in terms of the magnitude of effect based on RV systolic pressures and RVH, pretty close to the effects that we'd see with the preclinical sotatercept molecule. And we saw this also at the level of inhibiting muscularization. This is after 1 week of treatment, and on the middle here, this is after 3 weeks of treatment. And we were a little bit surprised to see that the hemodynamic effects could even be seen even after 2 doses in 1 week. To understand mechanism, Ying looked at a number of published and unpublished data sets, looked at the Stearman microarray data, and looked at the Sagan single-cell RNA-seq data from several patients and controls, and then looked at our own transcriptomic data on microvascular disease treated with BMP9. And what she noted was that many of the genes that are common to pulmonary hypertension clinical samples and to the transcriptome of BMP9 in microvascular ECs overlapped with many of the targets that we've focused on both in clinical pulmonary hypertension and currently in development for the treatment of pulmonary hypertension. She did a little bit more comparisons of these different data sets and came away with CXCL12 as being a very consistent signal that was derived from endothelial cells as well as other cells, but was potently regulated by BMP9 and microvascular ECs and seen in experimental rat Sugen hypoxia disease as well as human disease. There were these other interesting gene products too, like endostatin and TSHZ2 that might be implicated. But CXCL12 is really well established as a potential driver of pulmonary hypertension. And here we see in this Sagan data set that CXCL12 is primarily expressed in several GCAP subpopulations, as well as arterial ECs and systemic venous ECs in pulmonary hypertension more so than in control cells. And it is a target of BMP9 in microvascular ECs, but you have to look at the right time. It's actually elaborated more after several hours, so it's not an early target, and you can see it's secreted after 16 or 24 hours. Well, it turns out, as we know from work in this audience here, that CXCL12 potently modulates the phenotype of smooth muscle cells and induces the expression of many contractile genes. So Ying did an experiment where she looked at the potential paracrine effects of BMP9 treated endothelial and smooth muscle cells that are grown in apposition. So BMP9 didn't do very much to the phenotype of smooth muscle cells at 1 nanomolar concentration, but when these cells were co-cultured, BMP9 induced a potent contractile phenotype switch in the smooth muscle cells. And this could be blocked by a CXCL12 neutralizing antibody or by CXCR4 inhibitor AMD 3100. Serum actually replicated this effect, and that could be partially blocked by inhibiting BMP9. Many of you know that BMP9 is very important in the pathogenesis of HHT. We did some HHT mimicry studies in a HHT-prone strain. This is the SVJ129X1 strain, which is very similar to the strains that were originally investigated for ALK1 and endoglin knockout to replicate HHT phenotypes. We show that potent BMP9/10 traps like ALK1FC and ACTR2BFC will elicit severe AVMs, which are manifest as digit bleeding within 2 or 3 months of treatment in this strain. And then you can also, if you have blinded observers, quantify the increased prominence of the digital vessels that are short of a bleeding episode, but we think are a marker of AVMs. And you'll see that sotatercept in green has a milder effect, and anti-BMP9 has a very similar effect, whereas anti-BMP10 is more potent, and then combined blockade of 9 and 10 are certainly very potent and give you the bleeding. So we also can draw from our clinical experience, and we know that all of the drugs in this family that were developed that blocked 9 and 10 very potently resulted in epistaxis within 3 to 4 weeks of their clinical trials. Sotatercept results in epistaxis and telangiectasias over several months, and that might be attributed to the fact that it weakly blocks BMP9 but potently blocks BMP10. So we wonder, following the history of luspatercept, which is approved for anemia, MDS, and myelofibrosis, it only blocks 10, has never been associated with epistaxis, that a pure anti-BMP9 blockade may be similarly safe. And it has cleared group phase 1 safety studies and is being advanced to a phase 2 proof of concept trial in pulmonary hypertension by Pfizer. So to summarize, there's a paradox of BMP9 function, clearly. We know that genetics implicated as a loss of function risk factor, and there's acquired loss of function in both portal pulmonary and hepatopulmonary syndromes. But then there's this very compelling evidence that inhibiting BMP9 is effective in preclinical models shown by other groups as well as ours. And we have some data I haven't shown here that suggests that the effector molecules SMAD159 and SMAD23 may both contribute to these pathogenic effects. Anti-BMP9 itself doesn't exert severe HHT mimicry in HHT-prone mice, and so we think this could be tolerable, but this has to be tested in the pulmonary hypertension population, which may be more prone to this than healthy volunteers. And so that's why a clinical trial is needed for both safety and efficacy. Ying Zhang did the bulk of this work, which was started, this project was started by Peiren Yang, who leads his own group in Beijing now, and many of the other members of the group contribute, as well as members of the Pfizer group and our collaborators at Brigham and University of Michigan. Thanks so much.
Thank you very much, Paul Yu, for this wonderful presentation and adding another intriguing puzzle to the field. We'll continue with the discussion, and I would like to give word to one of the panelists.
Francis, if you have a question. Otherwise, I just wanted to ask Paul, I know you have described that before at the conference, but just for this audience, maybe somebody missed it. The whole like discrepancy or the controversial let's say results. You had a nice slide at the ATS meeting. If you could just highlight the technical reason behind those discrepant results in relation to the BMP9 experiments.
Yeah. Okay, happy to. So in the session, somebody asked how it was possible that BMP9 had such nice therapeutics effects before in these models, and we're seeing a very therapeutic effect of the opposite of inhibiting. How can both be true? So we actually, for the first part of this project, spent a year and a half trying to replicate the Nature Medicine experiments from Nick's lab. We contributed to that by using the same material that he used from Pfizer at the time, and saw that there were therapeutic effects of the protein in a Sugen hypoxia mouse model. So that was maybe the 5th or 6th model that was included in that paper. Pfizer generated new material for us that was validated by their protein chemists, and this material was felt to be very normally folded, complex protein with the pro domain as it circulates, and we administered it at doses that were similar to what were in the Nature Medicine paper, and we could not replicate those results. There were no therapeutic effects when we tried to repeat the study using the new protein, also made by Pfizer. This was true in the monocrotaline model, with no impact on hemodynamics, RVH, or remodeling. It was also true in a Sugen hypoxia model. Maybe there was a very mild effect, but nothing like the kind of really potent effects that we're seeing in the original studies. And this was true whether we used 2x of the published dose or 50x of the published dose. So we really went to some pretty great lengths to try and replicate the studies, and there were no impacts on remodeling in the Sugen hypoxia. We even did RNA-seq to see if any genes were regulated by this BMP9 protein. And actually, there were no transcriptomic changes in the animals that were treated with BMP9 compared to Sugen hypoxia by itself. We took a look at the protein, and this is what we found. Number 3, lane 3, are the non-reduced and reduced lanes representing the protein that was generated for the Pfizer 2015 Nature Medicine studies. And then lanes 1 and 2 in both gels are the non-reduced and reduced protein. The new protein has both the pro-domain and the individual monomers and dimers when they're unreduced of the mature BMP9 protein, whereas the original protein used in the 2015 studies had a predominant non-disulfide linked monomer species of the BMP9. We think that a high fraction of non-disulfide linked mature BMP9 could act as a competitive inhibitor of signaling rather than as an agonist of signaling. And we draw from other literature where people have used recombinant BMP ligands or GDF ligands and thought that they saw agonist effects in their animal models, but were probably seeing the effects of poorly linked protein acting as competitive inhibitors. This is from the GDF11 literature. So, Saban, I think that was your question. Hope that clarifies where we stand on this.
Yes, thank you so much.
Perfect. Thank you for this very nice and elaborate illustration of chemistry, which makes it probably even more complex. And with that, I would like to thank Paul and switch to the last speaker of this evening, Sue Gu from Anschutz University, who will talk about sex differences in cardiac macrophages in pulmonary hypertension. Please go ahead, Sue.
Great, thank you. Can everybody hear me okay?
Perfect.
Great. So I'm going to wrap up the session with a little bit of a beginning story about sex differences in cardiac macrophages and pulmonary hypertension. So directly addressing the question Saban keeps asking everyone about whether there are sex differences, and this is the main thing that we're interested in. So starting with, there's been a lot of setup about the lungs and pulmonary hypertension. What I'm interested in is the right ventricle. And the reason for that is that, as this audience probably knows, mortality in pulmonary hypertension correlates with right ventricular function, actually, and not directly with PVR. So this study from Verdonk shows that in baseline studies, if you stratify patients by PVR, there's actually no significant changes between these two parts of the cohort in terms of survival. But then if you stratify by right ventricular ejection fraction, there is significant decrease in survival in patients with already baseline decreased right ventricular ejection fraction. And then if you further stratify by both right ventricular ejection fraction and PVR, the population that comes out with decreased survival is the patients with right ventricular ejection fraction, whether with elevated PVR or decreased PVR. And then this holds true even in response to PAH therapy. So further along in this paper, in comparing patients that survived versus non-survivors with pulmonary hypertension, the non-survivors were mainly different in that the change in the right ventricular ejection fraction over time was significantly decreased. The other reason that we're interested in right ventricular failure is that, as we know, pulmonary arterial hypertension is a rare form of pulmonary hypertension, but all other forms of pulmonary hypertension, including those that are much more prevalent, such as PH associated with left heart disease or PH associated with lung disease, are all associated with right ventricular failure and can lead to death from RV failure. Another point of background is that in the we know that females are more prone to developing pulmonary arterial hypertension, but due to this this effect called the estrogen paradox, although females are much more likely to have pulmonary arterial hypertension, both PAH and PH patients, females exhibit better survival. So in this PAH cohort, as well as in this general PH cohort that was based on a VA study by Corey Ventituolo, there is significant improvement in survival in females with PH compared to males with both pre and post capillary forms of PH. And we've also known that preserved RV function in females contributes to better survival in pulmonary hypertension. So females tend to have improved right ventricular ejection fraction compared to males. And in this study published in Chest in 2014, 40% of the survival difference between males and females was actually attributable to this improvement in RV ejection fraction. So the main questions I want to address with my research are, what are the mechanisms of right ventricular cardioprotection in females with PAH? And what roles do macrophages play in RV protection in females? So Andrea Frump and Tim Lamb, when he was at the Indiana University, started to establish and elucidate this story. And now over the past 10 years have established this important role of estradiol in improving right ventricular function in human RV and in animal models of PH. So in this one graph, they illustrate that female Sugen hypoxia PH rats exhibit better RV function than males, that overectomy worsens the RV function in Sugen hypoxia females, and that estrogen or estradiol repletion in overectomized females and males, which is not shown on this graph, actually improves the right ventricular function. This is unpublished data from Vijaya Karor when she was working in the Lamb lab, showing that overectomy increases while estrogen or estradiol repletion attenuates the expression of inflammatory markers in the right ventricle in rats with Sugen hypoxia PH. So stepping back further to just establishing why are macrophages important to study in the right ventricle. And first of all, we know that macrophages are the major immune cell of the heart, and that we've established in my previous work when Brian Graham's lab was at the University of Colorado that right ventricular macrophage subpopulations expand in multiple types of pulmonary hypertension. So here's showing that right ventricular macrophage subsets, the IM1s and the IM3s, which Raul really nicely demonstrated that those populations with flow cytometry in his presentation, they expand with in mouse hypoxia pH. So by 24 hours and even 48 and 7 days of hypoxia, there's an expansion of this IM1 population, as well as this expansion of the IM3 population, which is mostly monocyte derived in mouse hypoxia pH. And then further, we looked using parabiosis studies where we had one mouse with and one mouse without pulmonary hypertension, and using parabiosis, we're able to tell which macrophages were derived from the residence mouse versus the partner. And so in this paper, we showed that this particular IM3 population had significant increase in the circulation-derived macrophages in response to pulmonary hypertension. And then we also went ahead and wanted to establish what role do macrophages play in the right ventricle in pulmonary hypertension. And so in this experiment, we did macrophage depletion using clodronate and showed that this improved the contractility as well as the RVP8 coupling in the more severe form of rat Sugen hypoxia pulmonary hypertension. And that the macrophage depletion actually resulted in the significant decrease in ECM collagens and collagen-related proteins. So in this more recent data that I'm presenting, I performed fixed single-cell RNA sequencing of the mouse heart in normoxia, 3 days of hypoxia, 7-day hypoxia, and 21-day hypoxia. And we included both female and male mice at each of the time points. So we did fixed single-cell RNA sequencing after after separating the RV from the septum and the LV and enzymatically digesting and generating a single-cell suspension. And we found that there were clusters of macrophages where, as Raul also demonstrated earlier, there's this CCR2 positive population of macrophages, which is primarily the monocyte-derived peripheral circulation-derived macrophages. And then over here, there's a big cluster of more resident cardiac macrophages that are the MHC2 high and the TLF or the FOLR2 expressing macrophage population. We were able to separate the male macrophages from the female macrophages using Y-linked genes in our data set. And in comparing the male and female macrophages, what was really surprising is that even at baseline in normoxia, there were significant differences and increased expression of ECM-related genes in the male macrophages. So collagen 1A1, fibrillin, LOXL1, collagen 6A1, and collagen 6A2 were all up regulated in the male-derived resident TLF macrophages. And then in hypoxia, there was a further separation of gene expression in RV macrophages. So by 3 days, there was downregulation of this group of genes as well as upregulation of these genes that are related to ECM regulation. By 7 days, there was still persistence of some genes in the right ventricular macrophages. What I'm not showing is that actually by 21 days, the changes were normalized, so there were no longer detectable differences in male and female macrophages. So when we put these genes into an IPA to look at the differences in canonical pathway expression, there was one predicted downregulation in male derived cardiac macrophages of the wound healing signaling pathway, but there were predicted differences in activation of MMPs, extracellular matrix organization, but in an unknown direction in the male derived macrophages. So just a summary of this kind of early data from comparing male and female macrophages and summarizing the background that we know about sex differences in the right ventricle. So we know that female sex and estrogen receptor signaling confers superior RV adaptation in pulmonary hypertension. We've established previously that right ventricular macrophages play a role in ECM remodeling and experimental pH. And that right ventricular macrophages, according to this preliminary study, have sexual dimorphic gene expression, both at homeostasis and in experimental pulmonary hypertension. MMP activation, ECM organization, and wound healing pathways exhibit significant sexual dimorphisms in mouse cardiac macrophages. And so now my future directions are the subject of my K08 proposal, where I'm investigating the interactions of female sex hormones, so 17-beta estradiol, with resident macrophage expression of ECM organization and regulating genes, as well as expression of cytokines that may potentially be involved in fibroblast activation. And so through both direct and indirect through fibroblast interactions, what the role is of the RV resident macrophages in the ECM collagen content and collagen cross-linking, and eventually resulting in changes in contractility and RV stiffness and diastolic dysfunction in the pressure overloaded right ventricle. And my hypothesis is that the female sex hormones modulate the expression in right ventricular macrophages of genes that drive fibroblast activation and regulate ECM organization, and that results in these sexual dimorphisms that we observe in right ventricular function. And so our goal is to understand the biologic mechanism of cardioprotection in females via the immune system in decompensated RV failure in human and animal models. To define the mechanisms of macrophage fibroblast crosstalk and macrophage ECM interactions in the RV. And then another future direction is I'm passionate about understanding the effects of menopause and the loss of estrogen signaling on RV macrophage programming and therefore on cardiac function. So I'd like to acknowledge the funding that has enabled this work. I was the recipient of an ATS Early Career Investigator Award in Pulmonary Vascular Disease, and then more recently an award from Team Phenomenal Hope, the Phenomenal Impact Fund for Global Research. My mentors, much of this work was done in Brian Graham's lab when he was at University of Colorado, and he's now at UCSF with the help of Raul and Michael Lee, who are still in his lab. At University of Colorado, Kurt Stenmark is my main mentor, and then as well, National Jewish Health, Tim Lamb is a major mentor who's helped with my exploration of sex differences in RV function. So with that, I will take any questions.
Thank you very much, and for this wonderful story. And yeah, to kick off the discussion, I would first like to switch to the panel. There must be any questions from these panel members, I think.
Yes, very nice presentation. Really intriguing story. So I understand your choice of looking to the macrophages, but will you ignore the other cell types, inflammatory cell types, or do you expect also their sex difference? Yeah, will you take it along in the future?
Yeah, I think all great questions. So Claudia, Mikael, and I did some of the single cell work together. So we're going to look at the dendritic cells in the heart as well. And of course, there's other cell types in the I did single cell sequencing of the entire mouse heart.
Yeah.
So a preliminary look at the fibroblasts actually showed no sex differences, which was really intriguing. But definitely we have the ability to look at all the different cell types in the heart.
And then
you go. No,
Go ahead.
you go.
No, your thoughts shouldn't be blocked. Go ahead.
Yeah, another small question. So you showed all the data on the right ventricle, but you also had left ventricle and septum. Did you see anything there, or did you only see things in the right ventricle happening?
In all macrophages from both the right, left, and septum, there were sex differences. I haven't looked at all of the other cell types yet in the left ventricle and the septum. And then other questions that we're going to explore with this data set is just RV and LV differences in general over the course of hypoxia. So a lot to explore with this one data set, fortunately.
Yeah.
And I'll just echo the question of my friend and colleague Lin Deng in the chat. We're just curious why you started looking at the hypoxia model for the RV. I could argue why, but we want to hear from you because you look at like day 1, day 3, day 7, something like that, like early time points. But then the RV function is not really impacted, and even the pressure overload is not there. So I'm just curious about the choice of methodology here.
Yeah, this is definitely just a starting point. So I'm working on developing the mouse PA banding model here at University of Colorado. Some of our collaborators, like at a speaker cotter at Stanford, have this, have the PA banding model well worked out at her lab. So we're trying to implement that here so that we can do parallel studies in a more severe model of PA banding RV failure. And I think that the other reason why choosing mouse over rat is simply that the markers are much more accessible in mouse in terms of looking at different macrophage subsets and macrophage recruitment. And those antibodies and those systems are just not well set up for the rat. So we have been working on that and developing a 10 or 12 color flow cytometry panel for the rat for rat cardiac cells, but still in terms of just subsets of macrophages, it's just not as well established. And in terms of targeting specific genetic modifications in rat, that's also just much more limited. But we definitely plan on exploring because each different type of pulmonary hypertension is heterogeneous or potentially heterogeneous in terms of the cellular differences in the heart. We do want to make sure that we confirm these findings in multiple different models and species of RV heart failure, and also using human tissues, which thanks to Sebastian Bonnet's group at University of Laval, we have human RV tissues that we're going to look at macrophage subsets in as well.
Yes, perfect. And with that future directive, I would like, or perspective, I would like to finish up. I think we are running already some minutes over time. And I really have enjoyed the session. I really want to thank the speakers for giving us such a broad picture of pH today, ranging from the right ventricle to the vasculature and from parasites to cardiomyocytes. So thanks a lot for the speakers. Thanks the panelists for the lively discussion. I think there was plenty of discussion and could be more. I hope that Sony Pulamsetti got through and is not still stuck in public transport. And I would like to end up with thanking Samantha Carpent for facilitating this wonderful webinar. I think we had a lot of attendance, so thanks very much for promoting. And I would like to ask Roberto, would you like to say anything about the next webinar from the PC Journal Club?
Thank you, Georgian. Next week on Thursday, it will also be at 2 p.m. Eastern Time. We're having the second part of Best of ATS. It will be the clinical discussion of some of the best abstracts. So next week, Thursday, 2 p.m. Eastern Time, please come and join us.
With that, I would like to finish up, and thanks all for attending. Have a good evening or a good afternoon.
Thank you.
Thank you.
Thank you. Bye.
Thank you.